My name is Ed Conway, just in case, and I'm welcoming you to, I think it's the 6th Center for Blood Research Day, really to celebrate the summer students of their hard work through this summer, as well as to gather everybody together today. So there are many people to thank. I really want to start off by thanking the sponsors, many of whom actually supported this day, or students throughout the program. So that includes CSL Behring, Pfizer, Grifols, Biogen, New England Biolabs, Bayer, Canadian Blood Services, Octapharma, Macropharma, the Sheldon Neiman and Linda Vickers Hematology Endowment Fund, and the Neil Mackenzie Memorial Fund. Now there are many others who are to be thanked, particularly the supervisors and the technicians and research associates and postdocs and all those other colleagues and friends who have helped tolerate these, help these summer students through the summer and getting their work done, as well as to set up today. But nobody warrants more thanks really than Hannah and Anna, who have really put their guts into setting this up each year. This is really the last couple of years I've really sat back and just come up here and then presented. And Amar, a more recent addition, has really been instrumental, and you all know Amar because she's really been involved in the student program particularly. So the day, I'm going to really go quickly because we have lots of things to do here. You'll have the students will be presenting 3 minutes each. And then we'll, at the end of that, which will take a little while because I think we have 26 presentations, and then there'll be the Neil Mackenzie Mentorship Award, and you'll hear a little more about that after. Is Dieter here? I hope so. Okay, we'll manage. And then we'll have a keynote speaker, Andrew Treitz, from the Marine Mammal Research Unit, which will be spectacular. And then we'll move over to the open bar outside in the atrium, where there'll be posters and poster judging. At 4:30, President Ono will appear for half an hour. It's only his second day of work on the job, so we're really honored that he'll be there, and he will be presenting the award for the oral presentation. And that'll be followed in or admixed with the barbecue and various other awards. Sorry. Okay, now you may wonder why I'm wearing this bow tie. I never wear a tie. Okay, so student presentations. This is sort of a picture of what you are all looking like now. So it'll change. Once you're up here, or once you're sitting down after you've given your talk, you'll be nice and cool. So, 3 rules. 1, do not look back. So don't go like this because nobody can see you and the video's running, and when you want to look at it later, you'll only see the back of your head. Okay, so you're going to face forward. You have a big fat pointer I've given you if you want to point, but you're really not it's not necessary. And there's no clicker, so you're going to have to use the keyboard to advance your slides. There will be a target of 3 minutes. At 3 minutes and 30 seconds, you will hear this sound. Okay, that's your warning sign, all right? And when it goes twice, you stop and leave the stage, all right? That'll be 4 minutes, all right? Way over time, but I know that none of you will go over time. That's what'll happen. At the end, during your so-called presentations, the students will score themselves, and I recommend you score yourself very highly, okay? And there will be, and based on that, at the end of the presentations, they'll be collated, and we will figure out who gets the best oral presentation, which will be presented, as I said, by the president. Okay? Any questions? Good. All right, so we're ready to go. Amar.
Do I just start?
You just start.
Okay. Hello everybody, my name is Amarpreet Graywal and I'm the Education Program Assistant here, or you may know me as a girl that sends a lot of emails. I'm here for my 8-month co-op term under the Faculty of Arts. The CBR mission statement, in pursuit of health through research in blood and blood-related processes, plays a role in why I do what I do. Even though this statement may not directly apply to me as I'm not in the forefront of research, however, each day I come to work with the goal to help advance this mission by contributing my efforts to offer a multitude of opportunities for our next generation scientists. This position has allowed me to take on things I really enjoy doing, which could be divided into 3 categories: awareness, student group support, and event planning. For example, to raise awareness about our center, I have developed the CBR brochure and the CBR banner, which can be found outside near the podium. So definitely check that out. And I am currently working on a social media strategy for outreach purposes and to attract a larger audience. Furthermore, I have implemented a project to capture the daily lives of CBR members. This not only recognizes the members, but it provides an insight for current and prospective students, staff, and faculty members of what life is like in the lab or in the office. It's also a great way for the patients and the public to stay connected with these scientists. As a psychology and sociology student, I have noted that in this high-stress academia environment, it is very difficult to maintain a work-life balance. Hence, I'm working on a mental health initiative to promote mental well-being through a series of workshops and activities. To get started, I have implemented a resource board in the pod with a list of resources available on campus for students and staff. Moving on, the CBR offers a lot of educational programs for academic excellence and applied opportunities, such as the Summer Studentship Program or the Health and Wellness Committee. Even though most of these are student-led, my primary role is to maintain communications and generally overlooking the groups and providing assistance whenever needed. The skills and knowledge I have gained over the past 4 months have provided me with the segue opportunity to plan my biggest project yet, the World Thrombosis Day Symposium on October 13th, which is a public education event. For example, by coordinating and responding to the industry tours by the summer students, it has allowed me to step outside of my comfort zone when it came to communicating with vendors to support or sponsor the event, which basically means getting discounts. Likewise, by organizing small-scale events such as the CBR seminars and the workshops, it has allowed me to use my time more wisely and be more organized by breaking up larger tasks into smaller ones. Overall, it's been a pleasure to work alongside and get to know many unique individuals of the center. So if you would like more information about the education programs, check out the poster outside in the atrium. Thank you so much.
Okay, so Carson, going forward, when you're finished your talk, just hit the next slide. The next person will come right up. There's a microphone here, so don't wander too far because they'll lose your sound. Okay? And again, the screen is there. Sasim is up there. Okay?
So good afternoon, everyone. My name is Carson, and I'm currently working in the Divine Lab under CBR's sponsorship. So I'm sure we're all familiar with the fact that blood components are vital in today's society, namely through surgical procedures. And it's just as important to protect our recipients as we protect our donors. So as all the CBR students know, thanks to the efforts of Dr. Conway in arranging for us to visit Canadian Blood Services, there are currently 2 protocols set in place to increase blood safety. The first, we have a oral screening of our donors to see if they've been to at-risk areas. And the second is that we actually test the blood for specific pathogens. However, these risks still exist as sometimes tests can't detect pathogens if they haven't replicated enough, so there's a detection limit, or that there might be unknown or unexpected pathogens present in the blood. So my project over the past 4 months has been to work with pathogen inactivation technology, where we activate a photosensitive agent using high energy UV light, and the photosensitive agent will then enter into the functional groups of nucleic acids and cause the pathogen unable to replicate. Now, there is a trade-off between blood safety and pathogen inactivation because, as we all know, platelets are a nucleate, but they still contain mRNA to create a protein throughout its lifetime. When pathogen inactivation technology inactivates nucleic acids, it will also affect mRNA within platelets. So my project has been to look at the effect of pathogen activation technology on these protein synthesis. So what we did was, using differential centrifugation, we separated the platelet components into three fractions, namely ones enriched for the membrane cytoskeleton, the cytoplasm, and the actin cytoskeleton. And after we get these fractions, we can run these fractions on an SDS gel and then immunoblot using specific antibodies to track the passage of proteins within the platelet. So here in the first example, you see RAP1, which is the GTPase. They initially start in the cytoplasm of the protein of the platelet. However, over time and after pathogen activation, they will enter into the actin cytoskeleton. Just as, say, Moesin, for example, they will travel throughout the platelets throughout their lifetime. And so after we get these immunoblots, we will compare it to the physiological health of the platelet during that time point, and then see how this inactivation technology will affect the platelet's health and whether or not the protein synthesis is damaged. So in summary, fractionating proteins using differential centrifugation improves the understanding of protein dynamics within the platelet. And upon this pathogen activation treatment, the GTPase proteins will move around inside the platelet, and it's this moving around that affects the platelet's health. And so further studies are currently necessary to determine the molecular mechanisms after pathogen activation technology. Thank you very much.
Good afternoon, everyone. My name is Andrew. I'm from Dr. Brahm's lab. Over this summer, I express a mutant enzyme. Cathepsin K is an enzyme expressed in osteoclasts and involved in bone resorption. So we can use it if we block it for the treatment of osteoporosis. Here we see mouse and human CAT-K superimposed on each other, and they're very similar. But one difference between the two is that mouse cathepsin K does not degrade collagen, as seen here, whereas the human one does. And the difference between the two is end-link glycosylation, found only in the mouse and not in the human. So, for my project, I introduced a glycosylation site into the human mouse cathepsin K, which normally does not have the glycosylation present, and evaluated its effects. So the difference between the two amino acid sequences of the mouse and the human is seen here. One difference that's pertaining to this project is at position 100. The mouse NAT is a glycosylation site, whereas the human NPT is not. So if we fast forward 2.5 months, the enzyme is made. So I purified it using 2 columns, and the mutant is seen here, and it's a bit bigger than the wild type, suggesting that the glycosylation was successful. And to further verify this, I used the glycosidase reaction to remove the glycosylation, and seen right here, the treated one is a little bit smaller than the untreated one, again further suggesting that the glycosylation is there. So now we have the enzyme, we compare it to the wild type in terms of the enzyme kinetics. Both the wild type and the mutant had relatively similar affinity for the substrate. The efficiency of the mutant is a bit lower than the wild type, but the stability at room temperature for both the enzymes is relatively the same. And here we see the 3 different substrates that I evaluated. So for the collagenase activity, we see that the human degraded the collagen, whereas the mutant, with the glycosylation present in an exocyte 1, did not degrade it as well. The gelatin activity was left unaltered, both degraded very nicely, and the elastin was hindered. As we see here, the wild type degraded far more elastin than the mutant. So in conclusion, the introduction of glycosylation in exocyte 1 did not affect gelatin degradation, but hindered collagen and elastin degradation. Thank you very much.
Hello everyone, my name is Andrew from Divine Lab, and I hope we all know that platelets help support coagulation, and this is important in clinical transfusion. But they also generate microvesicles during storage, and these microvesicles, called PMV, have variable levels of procoagulant activity based on the mode of platelet activation. Storage as one of these modes implies the variable of time, yet the effect of time is unknown for the procoagulant activity of PMV. So in order to find the answer to this question, we must isolate the PMV, and we do this through two separate centrifugations. We start with a platelet concentrate containing plasma, PMV, and platelets. Then we separate the platelets and then the PMV afterwards. This leaves us with 3 different samples which we take through 3 different assays. First, we measure the platelet count using a hematology analyzer. Then we measure the PMV count using a flow cytometer. Finally, we measure the thrombin generation of these samples, which we define as procoagulant activity for the sake of simplicity here. To find the procoagulant activity of each of these subcomponents, we can use subtraction between these different samples. Once we do so, we find that the total activity of platelets increases significantly over time, mirroring the increase in the total BC, which is represented by the blue and purple bars here. The yellow bars represent the plasma, which is the background noise of the assay, and the orange bars represent the PMV. We can see here that if we disregard the day 2 PMV with a huge standard deviation, there is an increase over time, albeit not significant. Looking closer into this, we find that the concentration of PMV increases significantly over time, yet the activity per particle does not. How we calculated this activity per particle is that we divided the total activity of the PMV by the count measured on the flow cytometer. This may explain the high values on day 2 because of the low values of PMV count on those days, leading to an inaccurately high data points. However, we can conclude that the total procoagulant activity of platelets and PC increases significantly over time. However, we discovered that the increase in total PMV procoagulant activity was purely due to the increased quantity and not to any change in their nature of their particles. Thank you.
Hello, I'm Cameron Henderson. I was in the Stranaca lab this summer working on insights into structural inhibition of undecaphenyl pyrophosphate synthase. So everybody knows bacterial cell walls have many varying components: peptidoglycan, lipopolysaccharides, wall-teichoic acids. They all have varying functions, but altogether they aid in the bacteria's ability to resist antibiotics. Shown here is a pathway for peptidoglycan synthesis, and one of the main components of that pathway is undecaphenyl phosphate. It's a lipid carrier that acts as a membrane anchor for the precursors of the many cell wall components. It arises by 2 means: de novo synthesis, whereby undecaphenyl pyrophosphate synthase, UPS, condenses 8 isophenyl pyrophosphate molecules with 1 farnesyl pyrophosphate molecule, or by recycling. So following polymerization of the precursors, undecaphenyl pyrophosphate phosphatase, P, takes it from its inactive form, the pyrophosphate form, back to its active form, the phosphate form. So why do we care? Co-inhibition of UP-S and UP-P could be an effective alternative treatment for bacterial infections, such as methicillin resistance Staphylococcus aureus. So to put it in perspective, in 2013, 2 out of every 1,000 patient admissions in Canadian hospitals was due to MRSA infections, and then the year prior, 8.6% of patients with a clinical MRSA infection died. So recently in 2016, collaborators in the Brown lab at McMaster, they identified a compound called MAC0547630 as a potential UP-S inhibitor. I know it's a mouthful. So the problem with MAC is that it shows greater inhibition of Bacillus subtilis UP-S compared to Staph aureus UP-S, and of course the latter is the one that actually is pathogenic. So if we were to generate co-crystal structures of this compound with these proteins, it can give us an idea of how to develop compounds for treatment in MRSA. So after restriction-free cloning, cell culturing, affinity chromatography, and finally screening, was able to generate Bacillus up-S MAC co-crystals. Those were sent to the Advanced Light Source of Berkeley for data collection, and shown here is the structure solved at 2.9 angstroms. If we look on the left, there's a surface representation of it, highlighting the binding cleft of one of the monomers. With and without surface representation, we can see that there's binding or electron density within the binding cleft. It's not well resolved, but it likely suggests that it is MAC. In their paper, it had low nanomolar affinity to Bacillus up-S, as well as it's a very carbon-based structure, as you saw, so likely it's going to bind. However, since it's not well resolved, optimization of Bacillus up-S and MAC needs to be optimized in order to gather higher resolution diffraction data to solve structure with further resolved density of MAC. Also of interest is the apo structures of both Bacillus and Staph up-S, mainly because it'll give us an idea of how structurally it's changing when MAC's binding. But most importantly, solving the structure of Staph up-S in complex with MAC will allow us to determine the structural basis for the differences in selectivity and specificity for inhibition of Bacillus and Staph up-S. And then from there, structure-guided modifications of MAC can be done to increase its specificity and selectivity towards Staph up-S in the treatment of MRSA infections.
Hi everyone, my name is Cami, and I spent the summer working at the Strinadka lab. And I want you to imagine for a second that you're living in the 1920s. World War I just ended and you had surgery. You're feeling pretty good, at least for the first day, and then you look at your wound. It's red, it's swollen, and it's oozing with pus. The doctor comes in and says, well, you got a bacterial infection, but there's nothing we can do about it. I know we hate it when the doctors say that. But it sounds like something that could just happen in the 1920s, right? What if I told you this could be us in a couple of years? The truth is, with the emergence of antibiotic-resistant bacteria, like methicillin-resistant Staphylococcus aureus, or MRSA, we're running out of antibiotics that work. That is why I, as well as some of the other members of the Strinadka lab, are interested in taking a structural approach to understanding some drug targets for MRSA. Now, antibiotics normally target proteins involved in cell wall synthesis. Methicillin and penicillin that we're familiar with target penicillin binding proteins, which are required for cross-linking. I'm interested in looking at a protein that's further upstream of this pathway called HMG-CoA reductase, or HMGR. This protein catalyzes the reaction of HMG-CoA to mevalonate, which then undergoes a series of other reactions to form undecaprenyl pyrophosphate, which is a lipid carrier required for the transport of peptidoglycan monomers across the cell membrane. So to start, I expressed this protein in E. coli cells and then purified it using nickel chelation and size exclusion chromatography. We recently set up some crystal screens, so hopefully in the near future we can get some X-ray data and even a structure. It turns out this protein is quite cooperative and purifies well, as you see in the single band here in the SDS page gel, and a monodispersed peak and size exclusion chromatography. We recently got some preliminary crystals, which is pretty exciting for me, and we're currently working on optimizing them. So having said all this, why is this so important, and why are we so excited and so hopeful that HMG-CoA reductase could be a potential drug target? Well, it turns out that there are actually known inhibitors of HMG-CoA reductase in mammals called statins, which are currently approved, well, several of them are currently approved for human consumption. So we're hoping, and the goal is, that with an HMG-CoA reductase structure from Staph aureus, we can maybe use these statins as chemical scaffolds for the design of novel therapeutics. And then hopefully we won't regress back to the 1920s where Staph aureus or a Staph aureus infection likely equated to death. Thank you.
Hi, I'm Alana, and I spent the summer in Dr. Cote's lab researching the association between markers of immune and ovarian aging in the context of HIV. So previous studies have shown that women living with HIV experience menopause at an earlier age than the general population, and that immune aging has been associated with a loss in ovarian function. In our study, we used leukocyte telomere length and leukocyte mitochondrial DNA content as measures of immune aging, and we used menopausal status as a measure of ovarian aging. And so the objective of our study was to examine the association between leukocyte telomere, mitochondrial DNA content, and menopausal status in the context of HIV to further characterize ovarian aging in HIV-positive women. Our study included 288 HIV positive and 210 HIV negative women who were all above age 12 and enrolled in the Children and Women Antiretrovirals and Markers of Aging cohort. As a measure of ovarian health, we used menopausal status, and we took self-reported menopause status and confirmed it with a measure of follicle-stimulating hormone, which was done by ELISA. As a measure of immune aging, we used relative leukocyte telomere and mitochondrial DNA content, and we measured that using multiplex quantitative PCR. So our initial results confirm previous findings that women living with HIV experience menopause at an earlier age than uninfected women. And in looking at our markers of immune aging, we saw that both leukocyte telomere length and mitochondrial DNA content decreased with age in HIV positive and negative women. When we constructed a multivariable model taking into consideration all the demographics known to affect these markers of aging, immune aging, we found that past and current smoking status, older age, and HIV positive status were associated with shorter leukocyte telomeres, whereas menopausal FSH levels were associated with longer leukocyte telomeres. And in the mitochondrial DNA multivariable model, we only saw indigenous ethnicity and past smoking to be associated with increased mitochondrial DNA content. So in conclusion, we found HIV positive status to be associated with younger age at menopause, and we saw that leukocyte telomere length and mitochondrial DNA both decreased with age. In addition, we saw that HIV positive status was associated with shorter leukocyte telomeres and higher mitochondrial DNA content. And in a multivariable model, leukocyte telomere length was negatively associated with HIV positive status, but positively associated with menopausal levels of FSH. And we didn't see any of these correlations with either HIV status or menopausal status and mitochondrial DNA content in the multivariable model. So in summary, our results going back to the objective of our study showed that ovarian aging was counterintuitively associated with longer leukocyte telomeres, but we think that longitudinal studies will be necessary to clarify the relationship between leukocyte telomeres and menopause in the context of HIV. Thank you.
So hi everyone, I'm Evelyn from the McNaughty Lab, and I want to start off with a brief question. So how many of you guys know anything about inflammatory bowel disease, IBD? Well, okay, well, it's something that many of us are reluctant to talk about, but today I'll be talking about, how IL-22 may be a potential therapeutic against one of the profounding characteristics of IBD through a mouse model. So I want to begin with a brief introduction about IBD and IL-22. So surprisingly, millions of people are affected by IBD every year, predominantly amongst young adults and teenagers. Although the specific causes of the disease remains unknown, it is believed to be due to the colonization of pathogenic bacteria resulting in dysregulated immune response. So there's actually two types of disease, Crohn's disease, where you find the extent of inflammation located in the cecum, and ulcerative colitis, where it only affects the colon, which is in red here. So the disease is characterized by the depletion of the protective epithelium lining, resulting in sometimes very painful ulcers. So there's no cures available for the disease, unfortunately, as of yet, and many patients have to resort to surgery. But recent research have shown that IL-22, which is a cytokine secreted by a variety of immune cells, may aid in the regeneration of the epithelium by stabilization in IBD models, as well as aiding through upregulation the expression of antimicrobial peptides to further aid the immune response. So we investigate whether or not this is the case in our IBD models. So this is basically a 22-step method where we would start by infecting the mice with Salmonella to mimic the start of the disease. And then we'll inject the mice with anti-IL-22 to neutralize the effects of IL-22. And then we will dissect out the spleen and the cecum and perform CFU counts, tissue staining, and RT-qPCR, which I'll be showing you in the next slide. So for RT-qPCR results, you can see for RAG3 beta and RAG3 gamma, which are both antimicrobial peptides that are regulated by IL-22, to be significantly decreased in our anti-IL-22 treatment group in comparison to our isotype control, which is our negative control here. And for histopathology analysis, so L is the lumen, M is mucosal, SM is submucosal. So for our isotype control, we can see the nice VLI-like structure corresponding to normal epithelium, in stark contrast to the complete disintegration of that nice VLI-like structure in our anti-IL-22 treatment group. So taking the results together, we believe that gut epithelial integrity is actually compromised in our anti-IL-22 treated mice, which suggests that IL-22 may play a role in protecting against IBD. Thanks a lot.
Hi everyone, my name's Hannah. This summer I had the opportunity to work in Dr. McGilliver's lab and also to collaborate with Dr. Stranadka's lab here at the LSI. My project focused on the assembly of nanodiscs for the study of prothrombinase complex components. So I'm guessing that you've all had a cut or scrape at some point in your lifetime and that flow of blood eventually stopped. Otherwise you wouldn't be here today. We all have our blood clotting proteins to thank for this. Shown is a diagram of the coagulation cascade and all of the protein factors involved in blood clotting. During the final stages of coagulation, the prothrombinase complex assembles on a phosphatidylserine or PS containing membrane in the platelet plug. PS is a type of phospholipid. To study this complex consisting of factor 10A, 5A, and prothrombin, we will be using nanodiscs or nanoscale phospholipid bilayers, which are essentially artificial membranes. Now, you may be thinking to yourself, what kind of graphic is that? Does she A, not know what the protein complex looks like, or B, not know how to use Adobe Illustrator? Well, I can tell you that the answer to that is C, all of the above. While my Illustrator skills do leave a little to the imagination, our imagination is really all we have to go off of when visualizing the prothrombinase complex. The end objective of this project is to assemble the three components of prothrombinase onto a nanodisc to see the gross structure of this vital hemostatic mechanism or complex for the very first time. Firstly, to do this, we have to assemble a PS-containing bilayer or nanodisc and then test the ability of prothrombinase to bind to it using cryo-electron microscopy. So nanodiscs are composed of membrane scaffolding protein, or MSP, and phospholipid. The MSP essentially gives the disc its structure and supports it, while the lipids act as the layers on which the protein can bind. Once I did my nanodisc reconstitutions, I ran my samples over a gel filtration column. Now on to my results. I first expressed MSP from E. coli, and as you can see in the SDS page gel on the left, I had a lot of expression. I then purified my protein using a nickel column. Since some nanodisc protocols call for the histidine tag on the MSP to be removed after purification, I did so with a TEV cleavage, shown on the right. I then sent my samples for N-terminal sequencing to confirm my his tag removal. In my reconstitutions, I ran many different ratios of MSP to phospholipid and also used different detergents. Shown are the size exclusion chromatography results for just 3 of my nanodisc trials. Oh, but look at that, 3 minutes is up. So I guess to find out what all of these squiggly lines mean, you'll have to come visit me at my poster later on. Thank you.
Hi, my name is Harwood, and I've been spending the summer in the Stranatka lab. And so, I guess, based on statistics, I'd say there are about 50 of you currently carrying a deadly pathogenic bacteria in your body somewhere. Now, everybody stay in your seats. Before you guys rush down to the ER, I would like to add that this bacteria is probably not very harmful to you, probably residing somewhere in your nose, in your respiratory system, or on your skin, in a mutually benefiting relationship. However, for the hundreds of thousands of others, they're not as lucky, and this bacteria is not as friendly, usually causing infection, which could eventually lead to death. Now, most of you have probably heard of this bacteria, Staphylococcus aureus, or Staph aureus, S. aureus for the lazier like me. Some of you may even know of the increasing prevalence of antibiotic-resistant Staph aureus and the increasing need of novel antibiotics. Now, this is where my work comes in. So before I keep going, I'd like to clear everybody's minds. No, I do not work with moldy grapes, and those are not moldy grapes, but that's just my poor representation of Staph aureus. You guys can thank my artistic skills for that. So recently, it has been found that Staph aureus releases a set of toxins known as phenyl-soluble modulins, or PSMs for short. Now, these phenyl-soluble modulins are amphipathic short peptides that are able to insert into the membrane and form pores, which can eventually cause the cell to lyse. This mechanism allows the bacteria to escape from white blood cell attack and to cause the inflammation that is associated with the infection. So these PSMs are released by a protein complex known as PMT4. This protein complex is formed by 2 ATPases and 2 transmembranal proteins. So my work focuses on structurally and biochemically characterizing this protein. Through the summer, I've been able to express this protein in a 4 protein complex, hopefully resembling its natural state in bacteria. I've also been able to characterize its ATPase activity and to attribute this activity to each of its ATPase individually. So further work on this hopefully will allow us to provide novel antibiotics to the increasing need against staph infections. So thank you.
Hey everyone, my name is Jack. I work in the Rossi lab in the Biomedical Research Center, and let me get started. So I'm willing to bet all of you that every single person in this room has had a bruise. Put up your hand if you've never had a bruise. Great, I was going to call them up to the front and just kidding. Usually what happens, you get a bruise, the muscle that's damaged gets repaired completely. Within a couple weeks, it's like nothing has happened at all. Sometimes, in certain disease, this muscle repair goes wrong. Cells called fibroblasts start to lay down deposits of fat, connective tissue, and this is what we call fibrosis. Now, fibrosis can happen anywhere in the body, mainly in, heart, lungs, etc., and can stem from a variety of different diseases. Now, there are many key players, key cell types that come into play during muscular regeneration. This can include blood vessels, which include endothelium and pericytes, muscle stem cells, otherwise known as satellite cells, inflammatory cells, such as macrophages, and one interesting type of progenitor that under certain circumstances can either provide trophic support and help regenerate muscle, or they can turn into fibroblasts and start causing fibrosis. These cells are called FAPs, fibroadipogenic progenitors. Now, when muscle's regenerating, all 4 of these different cell types have a really complex and intricate orchestra of signaling that happens between them. When the signaling goes wrong, fibrosis can happen. In our lab, we study muscular regeneration and how it can lead to fibrosis. We found that FAPs and endothelial cells in blood vessels always keep a close proximity to each other. Therefore, our project focuses on the interaction between these two types of cells, what the signaling pathways between them might be, and how they might lead to fibrosis. What we do is we take both healthy mice and mice that simulate fibrotic disease. We inject their legs with a compound known as notexin, which is isolated from snake venom, which is not as deadly as it sounds. Basically, you inject it and it simulates a bruise. We then take those muscles and analyze them using flow cytometry, histology, RNA sequencing, and a new technology called CyTOF. Now, what we found is that there's one protein called angiopoietin-1, which acts on blood vessels, that's only actually secreted from FAPs of all the different cell types. So what we did is we took an inducible Cree system, we prevented FAPs from producing this angiopoietin-1, and we found that muscular regeneration is severely impaired. And we're continuing to characterize this interaction. So our research on the interaction between these two cell types can help us have a better understanding of the mechanisms behind fibrosis. And hopefully one day we're going to be able to drive FAPs towards their trophic support role and away from their fibrotic role. This can open up therapeutic targets in not only muscle, but in the lungs, the kidneys, the heart, and many more. So if you'd like to find out more, I'd love for you to come check out my poster later, and thank you very much.
Good afternoon, everyone. My name is Kimia, and I'm a summer student at the McNaghy Lab in the Biomedical Research Center. Today, I'm going to talk to you about a topic that has directly or indirectly affected every single person in this room. Yes, I'm going to talk to you about cancer, but don't worry, it's good news. This is a protein conserved between different species and part of the CD34 family. It is called podoclixin, and under normal conditions, podoclixin is present on podocytes, vascular endothelia, and specialized epithelia. But what about abnormal conditions such as cancer? Well, in a cohort study involving breast cancer patients, we found that high expression of podoclixin was positively associated with negative outcome. So we decided to investigate this a bit further, and we knocked down the podoclixin protein in a highly invasive human breast cancer cell line called MDA231 that naturally expresses podoclixin, and we found in an in vivo model that the tumor sizes actually decreased. Interestingly, when we looked at the lungs for metastasis, we also found fewer number of nodules. So although this is very significant, we still don't know the functional aspects of podoclixin. We know that the protein is important in the ability of tumors to metastasize, but how does this happen? How does the protein actually achieve this? To study this question, we decided to knock out the podoclixin gene, and this summer we used mutant constructs of podoclixin in order to try and find the domains most significant in cancer progression. We knocked out the podoclixin gene using CRISPR-Cas9 technology, and then we produced two different cell clones, B7 and G2. We then used mutant constructs of podoclixin, one without the extracellular domain, one without the intracellular domain, and one without the PDZ domain, which is the region that binds scaffolding proteins that act on the actin cytoskeleton. And we introduced them into our B7 and G2 cells. So once we had stable cell lines, we had to validate that our knockout of podoclixin, the wild-type form, actually worked. We did a flow analysis, and as you can see from the histogram, neither the G2 nor the B7 present any podoclixin. We then transfected our cells with our mutant constructs of podoclixin, and we decided to do a Western blot in order to detect the protein presence. As you can see, the B7 cells did produce the right proteins because of the bands. However, our G2 cells did not, meaning our G2 cell transfection with our mutant podoclixin constructs did not work. To conclude, we have shown that podoclixin is associated with a negative outcome in breast cancer patients. We also believe that podoclixin plays an important role in metastasis. At a later date, we will test in vivo which of the domains of podoclixin play the most significant role in cancer progression, and we will do so using our B7 cells and newly made G2 cells, and we will test them in a mouse model. Thank you for your time, and if you'd like to learn more, please come and see my poster.
Hello guys, my name is Kurt. This summer I had honor working with Dr. Tyler Smith at BGH. So warfarin, it's an anticoagulant that is commonly prescribed for atrial fibrillation and venous thromboembolism. Despite its efficacy, it's commonly associated with serious bleeding complications where 10 to 16% of the patients do experience. So in this case, we really have to find a good mechanism of reversing warfarin. And currently, PCC use along with vitamin K are the most effective mechanism for reversing warfarin. At BGH, historically, PCC issuance has to be approved by a pathologist. However, on March 11, 2014, BGH implemented the physician pre-printed order, allowing clinicians to bypass this approval process and give the patients the PCC as soon as possible. However, for off-label indications where it's not urgent or it's non-warfarin reversal, they still have to consult a pathologist. So our methods are really simple. We look at 2 years prior and 2 years post PPO introduction, and we compare them based on a number of factors. Here is a quick snapshot on the pre-printed order. Clinicians will have to tick off one of the indications, and if it's off-label use, then they'll have to consult a pathologist. And below that is the dosing guide for clinicians to select the right doses for PCC. Results. First of all, we did not see a significant reduction in turnaround time, which is not shown here, but surprisingly, we expect PPO to allow the clinicians to get the PCC faster, but that wasn't the case. Secondly, we look at the indications for PCC use. We found that there is an increase in indications for urgent surgery, and this really gives us the doubt that whether the surgeries are really urgent as defined as within the next 6 hours. So that's something we want to look further into. Lastly, we look at the appropriate dosing and INR correction. We see that there's an increase in correct PCC dosing, but however, there's a reduction in INR correction. So this really could be due to a lack of consultation from a pathologist. And so this, in combination of the same level of turnaround time for PCC, really gives a question whether the introduction of PPO really improves patient care. Thank you.
Hi, I'm Rora from the Overall Lab. Do you know that the protein S100A11 is overexpressed in many human cancers? It even influences the proliferation of cancer cells. With my research project, I wanted to find out how S100A11 regulates pro-inflammatory functions and how MMPs are affected. The Overall Lab studies matrix metalloproteases, MMPs. MMPs regulate cell proliferation, migration, and play a role in inflammation and metastasis. I have found out that my protein S100A11 is a substrate of MMP8 and MMP9. That means that S100A11 is cut by these enzymes. To determine the pro-inflammatory function of S100A11 and how this activity is affected by MMPs, I used 2 different immune cell lines. I used Th1 monocytes and raw macrophages. I stimulated them with my protein and they gave me TNF-alpha back. I detected that with the TNF-alpha ELISA. I also stimulated A431 cells. These are skin cancer cells from an old woman. I found out that with higher amounts of S100A11, the cell proliferation of my cancer cells increases. I even detected with Western blotting that my cells contain S100A11 and that they even secreted. But to which receptor does S100A11 signal? For that, I used special designed HEC blue cells. If a ligand binds to the TLR2 or TLR4 receptor, NF-kappa B is activated. That results in the expression of secreted alkaline phosphatase. SEAP shines in a bright pink color and you can detect it with a quanti-blue detection assay. All in all, we can say that S100A11 is a substrate of MMP8 and MMP9, that it has pro-inflammatory activities that is important for cell proliferation and that it signals to TLR2 and TLR4. If you want to know more about my protein, feel free to visit me at my poster.
All right, hi everyone. My name is Leo Yafet, and this summer I worked in the Castrop lab, and I was exploring the potential fibrinolytic activity of coagulation factor 13. So coagulation factor 13 is at the intersection between clot formation and clot degradation. And so at the end of the coagulation cascade, fibrin gets formed, and it forms a mesh, which serves as the foundation for our clots. And what factor 13 does is it cross-links this mesh and stabilizes the clot. Additionally, it incorporates these proteins into the clot that prevent degradation. Accordingly, factor 13 is also sometimes referred to as fibrin stabilization factor. So recently in the clinic, factor 13 has become more popular, and it's being used in patients that have factor 13 deficiency, or in patients with hemophilia who are experiencing acute bleeding episodes. So we wanted to explore the mechanism by which 13 is able to treat and reestablish hemostasis. So first we confirmed using a turbidity assay, that increasing 13 levels in a blood plasma clot was able to increase the time that it takes for clots to break down, aka the lysis time. So now establishing this, we hypothesized that it was the incorporation of these proteins into the clot that prevent degradation that allowed 13 to increase the lysis time of the clots. So specifically, we focused on alpha-2 antiplasmin. So we took plasma that was deficient in this protein that prevents degradation, and when we found that when we added and we increased 13 activity, that the clots will actually destabilize and lysis time decrease. So this completely contradicts its well-established role as a stabilization protein, and really shines light on our incomplete understanding of coagulation. So with this in mind, we hope to in the future explore the mechanism by which 13 is able to destabilize clots in the absence of alpha-2 antiplasmin, and also explore other potential conditions in which 13 is pro-fibrinolytic. Thank you so much for your time.
So one way we can treat tumors is to take advantage of the unique features of the tumor microenvironment aside from normal tissue. So tumors can cause cells lining our blood vessels to become leaky, which basically means molecules that are in the bloodstream can easily go into the surrounding tissue. Now this is a problem because it allows tumors to spread easily. However, we can use this to our advantage to deliver drugs directly to the site of the tumor. The problem lies with the therapeutics themselves. So a lot of the drugs that we develop have low solubility, which basically means we'll need more of it in order for it to be effective. And further, a lot of them can have toxic properties. Anyone who's taken medicine knows this from the mile-long list of side effects and warnings that a lot of the seemingly simple drugs come with. So this is where polymers can come in. Polymers are long chains of repeating units, and basically when they're conjugated to drugs, they can change the properties to allow for better distribution throughout the body or reduce the toxicities. Now a lot of polymers have been studied, but in science we're always striving for even better solutions, and this is where ultra-molecular weight hyperbranch polyglycerols come in. So besides being a mouthful to say, these polymers are repeated units of glycidol, and they show promise in therapeutics for having a low viscosity and low toxicity. But before we can use them in the body, we have to make sure that they're safe to use. So this was the aim of my summer project, to study the biocompatibility of hyperbranch polyglycerols. Or as I like to call my summer project, the summer I spent chasing people in the CBR for blood donations and literally giving my blood, sweat, and tears.
So yes, we did have to use fresh blood for these studies, and we used them to study coagulation, red blood cell health, and the immune response. And we did them through the following methods. We looked at the intrinsic and extrinsic pathways for coagulation, as well as clot formation and platelet activation. In addition, we also looked at red cell hemolysis, red cell aggregation, and complement activation. And we compared these novel HPGs to those of commercially available and well-studied polymers like dextrin, polyethylene oxide, and polyvinyl alcohol. So our results show that for both the commercially available polymers and the new novel HPG polymers, there was no effect on platelet activation, coagulation by either pathways, and hemolysis. However, the results get more interesting with aggregation. As we can visibly see, the commercial polymers aggregate significantly more than the synthetic HPG polymers. And this may be attributed to the low viscosity of HPG. Further, we see an increase in aggregate size with concentration for the commercial polymers, but not for the novel HPGs, indicating that HPG might be able to be used at a wider concentration range. So what we learned was that the novel HPGs are blood compatible and show reduced aggregation compared to that of the commercially available polymers. And these polymers are potential candidates for drug conjugation. So if you want to know the whole story, my name's Lily and I'm from the J. Kizakidatu Lab, and you can come visit poster 26. Thanks.
Hi everyone, my name is Massa and I'm from the Foster Lab. Could you all do something for me real quick? Can you imagine that for an entire day, you can't remember your own name or where your job is, or you can't recognize any of your friends or your family? That's really hard, isn't it? That's life for people who have Alzheimer's disease every single day, and yet there's no drug or cure that has been proven to work on these people. There are some drugs currently under development, and one of those drugs is called thiamine G, as you can see right here. Now, in healthy neurons of normal people, healthy people, there are these tiny proteins called tau proteins that are involved in the microtubules of the cell. In diseased neurons of Alzheimer's patients, these proteins are hyperphosphorylated, so they kind of stick together and form these aggregates within the neuron that blocks the signaling pathway. So the neurons slowly start to die right after that. But thiamine G has been seen to kind of decrease this aggregation process and slow down disease progression in mice who have Alzheimer's. Now, no one has ever looked at proteins other than tau within the neuron and see how it changes in response to thiamine G. So the question we asked was, how does thiamine G change the levels of different proteins within the cell? We wanted to look at proteins and we wanted to quantify them, so we used this method called SILAC, stable isotope labeling with amino acids in cell culture. You can tell why it has an acronym. So for that you would need cancer cells because they grow very quickly and they save you a lot of time. We used these cancer cells from a brain tumor in a 4-year-old girl, and we labeled some of those cells with a heavier version of some amino acids, and we create heavier versions of some proteins. You can call those cells heavy labeled cells. You can treat your heavy labeled cells with your drug, which is thiamine G in this case, and leave your unlabeled cells untreated. Then you mix these two samples and you give them to Nick if you work in Foster Lab, and he will run it on a mass spectrometer for you and give you a spectrum where you can compare the levels of different proteins based on mass differences. What we found was 1,051 proteins, 600 of them quantified, and only 5 of those proteins had changed in terms of their levels. And one of the key proteins that we found was really interesting is matrin-3 right here. As you can see, it's involved in another neurodegenerative disease called ALS. Now, according to our results, thiamine G has a very minimal effect on the proteome of the cell. So what's our next step in studying this interesting potential Alzheimer's disease drug? You can come check out poster 20 to find out what's next, and I'll be very happy to talk about it with you. Thank you for your attention.
So, hi professors and fellow CBR students. I'm May from the Carson Lab,
and my project was on the cross-platform comparison of DNA microarrays applied towards diagnostics of myeloid malignancies. So many of you are familiar with acute myeloid leukemia, and perhaps to a lesser degree, myelodysplastic syndromes, cancers in which blood cells are unable to form, rendering the patient vulnerable to infections, bleeding, and anemia. There's currently no cure for MDS, and AML has a cure rate of less than 30%. However, what we do know is that these diseases are caused by chromosomal mutations, and characterizing them can allow us to predict a patient's chances of survival and even potentially develop new therapies. So one of the mutations that my lab was particularly interested in looking at was a gene fusion, a hybrid gene that combines two previously separated genes. In this example, we have a gene fusion that is caused by a translocation. Gene B from chromosome 22 has now moved to chromosome 9, creating a gene fusion that is now made up of gene A and B. So what causes gene fusions? Gene fusions can be caused when the chromosome is inverted, deleted, moved, or duplicated, and their prevalence in AML makes them ideal diagnostic and prognostic markers. A famous example is the AML1-ETO fusion protein, found in 10% of AML cases. Patients with this mutation are generally said to have a good prognosis. So how do we detect gene fusions? RNA sequencing is particularly adept at identifying gene fusions that are caused by translocations and inversions. However, it has difficulty determining whether a gene fusion also involved a deletion or a duplication of a chromosome. And for this, you would need array CGH. So my project this summer was to look at 3 different array CGH platforms to determine which one will best complement RNA-seq-well and can be applied in a diagnostic setting. So the 3 platforms I looked at were AFI, Agilent, and OGT. What we found is that OGT was able to identify mutations not present in all populations of cells, be consistent, and not make false mutation calls like the other 2 platforms. To find out more about how I came to this conclusion, please come and visit my poster. Thank you.
Hi everyone, my name is Raymond and I work in the Chem lab. My primary studies focuses on dense granules and platelets. Platelets are one of the most numerous cells in blood and its main function is to help with clotting. Platelets contain over 300 active substances from 3 different types of intracellular granules, alpha, dense, and lysosomes. Certain agonists such as thrombin and collagen causes platelets to activate and make them secrete these granules. Recent studies have shown that different molecules of alpha granules have been differentially secreted. So my question is, are dense granules also differentially secreted? To test this, 2 different experiments were conducted. First, immunofluorescence to test the localization between alpha and dense granules, and a biochemical assay to compare the secretion rate of different dense granule molecules. For the immunofluorescence, the images for the alpha granules, which is PF4, and the dense granule serotonin was compared. You can see here that there are distinct staining, which indicate that they are located in different compartments. What's interesting is the immunofluorescence in the bottom. Both serotonin and polyphosphate are dense granule molecules. However, only partial co-localization, which is shown in yellow, is observed. This indicates that even though those 2 molecules are both in the same granule, they may have been packaged differently. For the biochemical assay, 2 different dense granule molecules, serotonin and ATP, were compared for secretion rate using strong agonist thrombin and weak agonist LPS. When the strong agonist thrombin was used, both ATP and serotonin achieved maximum secretion rate really rapidly. However, when the weak agonist LPS was used, the secretion rate for ATP and serotonin were shown to be different. Based on these results, different dense granule molecules may be released differentially, and this may possibly be due to difference in packaging. For more information, please check my poster. Thank you.
Hey, I'm Riley. I'm from the Conway lab, supervised by Tara Fernandez. CD248 is a transmembrane glycoprotein which is highly expressed in stromal and mesenchymal cells. Its levels are dramatically increased in cancer and inflammation. Mice lacking CD248 are protected against this cancer and inflammation. It would be suggested that strategies to combat these diseases would be to reduce the levels of CD248. My goal was therefore to identify factors that suppressed the expression of CD248 with the long-term aim of gaining new insights to treat inflammatory diseases such as arthritis, obesity, as well as cancer. To achieve our goal, we used an in vitro cell model. Several mouse fibroblast cells express high levels of CD248. We cultured them in the presence of different factors which we predicted would participate in cancer and inflammation. These were then harvested, lysed, separated by STS page, and evaluated for CD248 expression by Western blot. We screened many factors, but here is an example of our results. This is a Western blot with the molecular weight markers on the one side, top bands indicating CD248, and bottom bands indicating HSP90 for equal loading. The control CD248 band is from untreated fibroblasts. Coumarin is a growth factor which is responsible for adipocyte differentiation and metabolic function. Exposure of the fibroblasts to coumarin for 6 and 24 hours show an increase in CD248. Platelet-derived growth factor, PDGF, a growth factor that participates in perivascular and vascular function, had a minimal effect at 6 hours, but suppressed CD248 at 24 hours. And then insulin-like growth factor, which induces several differentiation and proliferation pathways, induced a biphasic response, causing CD248 to first go up and then go down. And overall, regulation of CD248 appears to be very complex, modulated by various factors. We plan on further characterizing each of these in more detail. The knowledge gained from these studies will hopefully be important in developing interactions in the CD248 pathway and may be of therapeutic benefit.
Hey everyone, don't worry, we're getting towards the end. There's only a few of us left. All right, so today I'm going to be talking to you again about HIV. We had a presentation earlier about it. I work over at the BC Center for Excellence in HIV/AIDS, where we do routine drug resistance testing for all of BC and a large portion of Canada, just not Quebec. You might notice that the second author here says Kimia, who presented earlier. So big thanks to her because she actually helped out with this study too. All right, so first I'll give you some background. In this study, we actually looked at 2 different cohorts from Swaziland. It's a really small country in Africa. The arrow actually points to where it is. It's just surrounded by South Africa and Mozambique. So it's a country of about 1.2 million people. And it's actually the highest burden country in the world for HIV. In 2003, it affected almost 40% of adults. At that time, the government decided to introduce HIV treatment called antiretroviral therapy. And last year, UNAIDS found that only 28.8% of adults had HIV. So it dropped by about 10%, which is a great stride. But as we know, when you start introducing drugs for bacteria or viruses, resistance starts to come up. So one problem is that these countries in Africa don't have access to drug resistance testing. So one thing they do is they send us the samples at the Center for Excellence, and we do the testing for them. So what we did in this study was we extracted the HIV RNA, amplified it by nested PCR, and sequenced it using Sanger technology. And what we found for our first cohort, which is from treatment experienced people, was that only 12.4% of samples managed to amplify. While this actually seems really bad, it's actually a good thing because most of these people are on treatment, which means they have very low levels of our HIV RNA in their blood. So it makes sense that so few of them actually get PCR products out of it. Out of the ones that did get amplicons, we sequenced them and we found that they fit into generally two groups. Samples that had absolutely no resistance, just completely susceptible to all HIV drugs, and those that were resistant to almost all HIV drugs. This is a sign that people are either failing their treatment or they're actually not even taking their drugs like we think they are. The second cohort we looked at was actually split into 4 separate groups. Without going into too much detail, you could basically split them into treatment naive and treatment experienced groups. We found that the treatment naive groups had very little resistance in them, but some patients did have baseline resistance, mostly to first-line drugs. For the patients that were treatment experienced, we actually found a much higher level of drug resistance, as high as 50%. In some samples, though, there was, again, absolutely no resistance. So in this separate cohort, we found similar results that show that patients might not be adherent to their drugs, or they might be very resistant. Yay. Yeah, so this is just a sign that we can do the testing for those countries and that their access to drugs is helping them. Thank you. Come see me.
Good afternoon. My name is Sabrina So, and I worked this summer with Dr. Raywin Brody on a chart review looking to see if serum ferritin levels can predict the development of metabolic syndrome in stem cell transplant patients. Now, why does the outcome of this very specific and complicated hypothesis matter to us? And the answer to that requires a bit of an explanation, starting with some good news. As advances are made in transplant technology and procedure, stem cell transplant success rates are higher than ever, and patient survival as a result is constantly improving. Good news, right? Not entirely. Unfortunately, when compared to the general population in the same age group, it's been found that these patients still fall behind in regards to life expectancy, and that is due to non-transplant related complications. One such complication is the development of something we call metabolic syndrome. Described in literature as a cluster of interrelated comorbidity, many of these common conditions are considered just as some extra baggage that comes with aging. However, actually developing metabolic syndrome, i.e. having 3 or more of these morbidities concurrently, is anything but harmless, and it does come at a price. Metabolic syndrome has been proven to increase risk of developing cardiovascular disease, stroke, diabetes, and all-cause mortality, something that I think the majority of us here today would very much like to avoid. Additional study has revealed possible linkage between high ferritin levels to the inflammatory pathways involved in metabolic syndrome. The actual role that ferritin plays in this is still relatively unknown, but this really piqued our interest as post-transplant patients often come away with high ferritin due to either the actual transplant being an inflammatory process or the many blood products that this population is exposed to. Which, of course, led us to pose the question in our hypothesis, whether or not hyperferritemia actually predicts metabolic syndrome outcome, which would be important information to know when trying to prevent it from happening. We sought to put our hypothesis to the test by heading to past patient charts to see if the patients who had high ferritin really ended up with metabolic syndrome. 578 patient charts were available for review and filtered through the following criteria. Unfortunately, only 80 of these patients ended up eligible for study, less than 15%, even though guidelines have been advising the regular testing of these serum markers since 2006. With these 80 patients, we found these results. In this graph, the patients are separated into two groups, patients with normal and elevated ferritin. From there, we analyzed the information we had on them, such as their cholesterol levels and their BMI. The blue bars are the patients we found to have one or more sign of metabolic syndrome, and the red bars are the people with none. Now, as you can see from the chart, there appears to be no correlation between ferritin level and the prevalence of metabolic syndrome signs. However, there are numerous caveats in this review that make this result a bit more inconclusive than it seems, such as the fact that the graph only really, as you can tell, look at the signs of metabolic syndrome rather than the development of metabolic syndrome itself. And the reasons behind this, as well as the other caveats of this review, I'll be happy to discuss in my poster. Arguably, the most important point that we found out during the course of this review is not only our hypothesis, proving our hypothesis, it was also the problem of physician non-compliance. And as a result, my supervisor and I are planning to launch a survey into, giving it to the physicians in hematology and VGH to try to shed light on some of the reasons behind physician non-compliance. Thank you.
Hello everyone, my name is Sargan and I'm from the Kazaki Dathu lab, where my project is to create a model for our vasculature. How cool does that sound? Tough crowd. Well then, glycocalyx, for those of you who don't know, is just a layer of protein and carbohydrates that's seen in the luminal side of the endothelial cell layer in our blood vessels, and its main purpose is to just protect our vasculature. However, the problem is that this glycocalyx is nowhere to be found in static cell culture conditions. If you look at the two images at the top of this slide here, you'll see the difference between the glycocalyx seen in vivo and glycocalyx seen in vitro, and the difference is massive. In vivo, we can see up to 900 nanometers of glycocalyx thickness, while in vitro, in static cell culture conditions, we only see about 30 nanometers of glycocalyx, which means that glycocalyx is pretty much irrelevant. So that's why we think that the original model needs a little bit improvement. And so how do we solve this problem? So we think that by creating environmental conditions that are similar to what the endothelial cells are experiencing inside our body, outside, we can actually create glycocalyx at levels that are similar to the in vivo levels. And we're using a microfluidic device to do that. So the picture of the microfluidic device is shown at the bottom of the chip, or bottom of the slide. And if you look closely, you can see that the chip has numerous channels, and each channel has 3 different regions. And that's important because having these 3 different regions with varying heights means that we can create 3 different levels of shear stress, which is just stress caused by flow of fluid, in a single chip. That way, we're able to create 3 levels of shear stress corresponding to shear stress in arteries, capillaries, and veins in just a single chip. So how do we do this? First of all, we have to design this chip in AutoCAD. And we get it 3D printed, and then we pour PDMS, which is a polymer, in this chip mold, and we let it cure, and following day we bond it to a microscope slide, and that's our chip. Following that, we seed HUVEC cells, which are endothelial cells, and we apply high shear stress and see how these cells react. So our results show that there's actually a significant difference in the amount of elongation that the cells go through, the ones that undergo shear stress, as compared to the static cells. And if you're not colorblind like I am, you'd also see the red in these pictures, and that red represents glycocalyx. And it's clear that the picture on the left that represents artery has a lot more red than the static, which shows that artery that receives the most amount of shear stress actually has a lot more glycocalyx upregulation as compared to static, which again validates that by increasing shear stress, we're able to increase the amount of glycocalyx and further validates our model. And in conclusion, I just want to say that this model, I believe, is a lot better, a lot more accurate representation of what's actually happening inside our body. And there's tons of future applications, which I am happy to talk about when you guys come see me. I'm poster number 3. Thank you.
Hi, thank you all for coming today. My name is Sujin Park, and I am a UBC co-op student currently completing a work term under Dr. Luke Chen in the UBC Division of Hematology. Today I'll be presenting a recently completed project entitled Overlapping Features of IgG4-related Disease and Lymphocyte Variant Hypereosinophilic Syndrome. IgG4-related disease is a fibroinflammatory condition with puffy glandular lesions characterized by lymphoplasmacytic infiltrate rich in IgG4 positive plasma cells. Lymphocyte variant HES is a unique subtype of hypereosinophilic syndromes characterized by an immunophenotypically aberrant T cell population, often with clonal TCR gene rearrangements. These two rare diseases share features such as eosinophilia and elevated IgE levels that makes distinguishing between them a diagnostic challenge. This case series aimed to compare the lab features between IgG4 disease and LHES in order to improve the ability to diagnose disease. We included 45 patients treated in Vancouver between July 2008 and June 2015 in Vancouver, with 30 IgG4-related disease patients and 15 LHES patients. We compared lab features including T-cell clonality and immunophenotype, IgG4 and IgE levels, as well as eosinophil counts. We found that elevated IgE levels were common to both conditions. 4 LHES patients presented with high IgG4 levels, and a subset of IgG4-related disease patients presented with eosinophilia. The most interesting of our findings was that while aberrant T-cell phenotype and clonality by PCR are defining features of LHES, these same findings were present in some IgG4-related disease patients. Ours is the first study to show the same T-cell immunophenotype and clonality findings in both LHES as well as IgG4-related disease. In terms of treatment, corticosteroids were effective in both groups, and for relapsed or refractory patients, rituximab was used most commonly in IgG4-related disease, and interferon alpha was used in LHES. So we found that in the differential of eosinophilia, IgG4-related disease must be considered as some patients present with eosinophilia and aberrant T-cell findings that may be mistaken for LHES. The markedly elevated IgE levels seen in both conditions raise the possibility of anti-IgE therapy, and the overlapping features suggest other potentially transferable treatment strategies between these two rare diseases. Thank you for your time, and if you have any questions, please come to poster 21.
Hi everyone, my name is Gary, and I work with Dr. Jackson at the Adult Hemophiliac Prophylaxis Clinic at St. Paul's Hospital. My project involves a review on the outcome measures for hemophiliac patients. And just to note that this clinic is very different and new versus other hemophilia clinics. Outcome, in this sense, means that the patient's attitude, behavior, and also wellness as a result of the intervention provided by the healthcare team. I'll give you some background on hemophilia. Hemophilia A and B are caused by deficiencies in the protein that is involved in blood clotting. And patients with these conditions usually have chronic joint bleeds, which causes the picture you will see here. So very difficult walking around and movements. So to prevent these bleedings, hemophilia patients are treated by self-injection at home, usually 2 to 3 times a week. And these can be very difficult and time-consuming. On top of that, they're usually very costly. So in this environment, collection of clearly defined clinical outcomes are necessary not just to justify public-funded treatments, but also to evaluate new treatment and increase patient satisfaction. Our objective is to examine these outcomes and assess feasibility of each for collection in a clinical setting. So a literature review and a chart review was conducted, and a list of current and potential patient outcomes are collected. We don't have time to go into each of these, but 5 major categories were identified, being clinical, functional, quality of life, lab pharmacokinetic measures, patient education, and economic outcomes. The outcomes highlighted in red are the ones that's currently being collected, and analyzing these outcomes, our result shows that if we look at the graph on the right, there are some outcomes that's consistently measured more than others, and the graph on the left shows that outcomes that's templated into the clinic notes are collected way more than those are not. So in conclusion, our findings suggest the benefit of templated clinic notes, and some traditional outcomes are collected more than those that deal with patients' quality of life. And consideration should be given to standardizing outcomes that are descriptive, numerical, and those collected with validated tools. The project is not yet over, and the next phase will include a focus group of clinicians to identify and negotiate key outcomes that should be collected in the clinic. Thank you.
So my name is Anika, and I'm a summer student at the Hancock Lab down by the Lower Mall Research Station. And so in the lab, I deal with host defense peptides, and these peptides are produced by any complex life forms and suggested by their alternative name, antimicrobial peptides. They're originally researched more so for their antimicrobial properties. However, they're also found to have immunomodulatory activities. So that's what my initial project consisted of doing. So I spent 3 months screening host defense peptides for anti-biofilm, such as MRSA biofilms, and also for immunomodulatory activities. However, the array that we ordered for derivatives of these peptides had low quality, so we had to put that project on hold. And then we moved on to a second project, which is a collaboration with the lab in Montreal. What we did is that we screened these host defense peptides for activity against plant pathogens, specifically fungi. So these are 4 of the 9 samples that they gave us. As you can see, there's a big difference in morphologies. It's really hard to culture them because they each have different growth conditions. But for today, I'll be focusing on the red strain. And so that's a Fusarium graminearum one. I just used that one because it was the easiest to grow, and it had the longest growth rate, which is 4 days. And also this causes rotting of the ear of the corn, also diseases in wheat and barley. So how the screening works is that you would take a full-grown plate, which is like 4 days old. You would resuspend the spores in potato dextrose broth, and then you would make your cereal dilutions of the peptide solutions and mix each one with the spore suspension. Lastly, you would spot the peptide and spore mixture onto the surface of the agar plate. And as you can see here, the dilution works twofold around, with the center being water, which is our control. So this is left at room temperature for 4 days. Our results are pretty good. I just put 2 of our peptides here just to show you that diverse range of activities. So here you can see that this peptide doesn't work too well, but again, you can see activity in inhibiting fungal growth. You see it in the higher concentrations. However, for the peptide over here, it's incredibly potent effect. You can see that there's no fungal growth whatsoever at any of the concentration tested. So this is the list of peptides that we did for the first screen. You can see that the one in the box are the ones that have really great activity. You can see that even below 16 micrograms per mil of the peptide, you can see inhibition of fungal growth. And so we hope to pick one of these peptides and further treat them against the other strains. What's also key to note is that these peptides are also shown to have antibacterial activity. The bacteria is Xanthomonas, and this is also a plant pathogen. So we were able to find a peptide that has both antifungal and antibacterial activity. It could be as a potential organic pesticide. And so that's the conclusion that we came up with, is that we have a lot of peptides that have really good activity. So again, we want to further move on with that. And also, we hope to repeat the screen. Actually, we did it yesterday, so we'll be seeing results on Thursday, and hopefully we can confirm our peptide results. Thank you.
Any other students who want to give a talk? Now's your chance. You've got 3 minutes. So first, I think we should let Victor, push that a few times because I think that it's fine. There you go. There you go. Good job. Okay, so amazing timing. So first, I think we should all give all these students a round of applause because they were really spectacular. The students have some work to do. First of all, you can scratch it. You don't have to put your name as judges, by the way. And second of all, when you're, because we don't really need to know. And when you've done your judging, if you can just pass them down to the end, and maybe Ed Prysdale will collect. We have something first, Andrew. Hold on. Is Dieter here? Dieter Brummer? Oh, good. That's our next order of business. So a few years ago, we lost a member of the Center for Blood Research, and Dieter's going to tell us a little bit about Neil McKenzie. But we've set up this mentorship award program in his honor, and we've had some amazing award, nominees for this award, and this is it's really a cherished award. All these nominees should feel very honored actually to be elected essentially by their peers and students. So Dieter, do you want to come up and tell us a little bit about Neil? I want to go the right way. Okay. The next one is the awardee, so don't push it.
So I don't know if there's a dress code today, so I have this thing here.
Yeah, it's a new thing.
It seems to be a general feature of all the male speakers. I don't have one. So I think Neil also never had one. So Neil was a postdoc of mine for 3 years. I mean, for 3 years ago he joined my lab, but he was only for 1.5 years in my lab. And he died now almost 20 months ago, tragically in a mountain accident. So he was really a mountain guy, always in the mountains, and crashed down, I think, an 800-meter ice fall with 2 other friends of him. So this was really a tragic loss for us. And Neil was actually sending me literally a day before a paper, not a scientific paper, but kind of a readout about Churchill and the general educational problems in the world. And I was making some notes, telling him we talked next day, and he never showed up again. So it was really a tragic loss for us, for all of us here. And he was really a good mentor. And so that's why I think we generated this award for young people who can be similar to him. And we want to present today, I guess, the winner. And you will know who is the winner. Evan Honey.
Is Evan here?
He's here.
So the last part of our sort of formal part of the presentation is our keynote speaker today. And Andrew, I couldn't decide which picture to show, so I showed both. I love it. When you go digging through the the annals of the internet and you find out here's a current CV of this speaker or professor, and then there's this picture of him when he's 20 years old. That's all I've got on the internet too. Or I try to do that, but Anna and Hannah make sure that doesn't happen. So as many of you know, each November, the CBR hosts a symposium in honor of Earl Davey, the discoverer of the coagulation cascade. And on the day after the symposium, we generally try to find something for him and his and the guest speakers to do, something special. And he's been to the Museum of Anthropology umpteen times, so he tried to steer away from that. So on a Friday afternoon, our standard meeting, Ross and I and the beer, Ross a few years ago said, Andrew Trites. He saw this, Ross had seen this spectacular movie on, and I don't know if it was on CBC or PBS, but it was the story of how the whale, the skeleton of the whale, was brought from Prince Edward Island to the museum here at UBC. And it was just amazing. So Ross contacted Andrew, and I think that they knew each other, and asked him, would you give a personalized tour to our guests, which he kindly did. And it was truly amazing. And if you can ever see this movie, and I'm not sure if it's still around, you should definitely watch it, because it's really amazing. So Andrew received a BSc in mathematics and ecology at McGill, and he'll have to explain the connection there. And then he completed a Master of Science and his PhD at UBC, and he's now a professor here at the UBC Fisheries Center. He's the director of the UBC Marine Mammal Research Unit and the director of the North Pacific University's Marine Mammal Research Consortium. I had to read that, sorry. As he writes, his research program is designed to further the conservation and understanding of marine mammals and resolve conflicts between people and marine mammals. And I hope he's more successful than it would be between people and people. And particularly focusing on seals, sea lions, and whales. And I love the title of his talk, especially for this audience, Expect the Unexpected from a Science Education. Andrew, and I just have to set you up.
The screen's reversed there.
I have a what?
So maybe just while we're getting set up, if you want to just stand and stretch, because I know you've been sitting.
Good idea.
So just stand up for 30 seconds, stand up. That way we're not focused on trying to get our slides here together.
That's a good idea.
And you have it backwards again. It's backwards.
Okay. How did I do it? What did I do?
Just duplicate the screens, I guess, if it's not going to work.
No, but how did I do it?
Where you found it?
How did I do it?
It was in, I think it was View, and then there was like an option you had to go and do, and then you click screens. Was it that right there, presenter too?
No, that'll just show.
That'll just bring it up.
I can't believe I do it this way. Set up shop.
There it is. Yeah, and then screen. And then you just,
I don't think so.
That's literally what we did, and then it worked. I don't know why.
So it's going to be reverse.
There we go. That's it. Yeah, we had the same reaction.
Don't understand what we did, Andrew.
Just drag this down here to make it
too.
slide bigger. Okay. Make it slightly bigger.
That good?
Yeah, that works.
It's all yours.
Great, thanks.
Thank you very much.
Does this work? Check. Yeah. Okay, well, thank you, Ed, for that introduction. As Ed said, that I did do my graduate studies here at UBC, and that photo he showed is, it's hard when you're a student and then you become a professor. So I had to go sort of incognito, so I grew a beard. And that way I could then sit at the table as though I was somebody brand new coming here to UBC. So I was impressed in listening to all of your presentations just about the diversity of studies and the incredible things you've been doing this summer. So I was thinking, so what did I do in my summer jobs? Well, I remember one of my first jobs is I got a job on a Soviet ship off on the Scotian Shelf, and I was the only person who spoke English on board all summer. and we were sampling zooplankton, and I learned how to eat borscht soup and speak just a little bit of Russian at the time. Another summer I worked in the Bay of Fundy, hiking in the mudflats sampling clams, and we're doing an assessment of what the effect would be if you build a dam to create tidal power, and how would the life survive. And I think the closest I got to some of the things you might be doing is I worked in an ecotoxicology lab where I got to take seagull eggs to look for PCB concentrations. I'm still very good at cracking eggs with one hand to open them up. But I have to say that, you know, during the time that I was doing my undergraduate degree, and here I'm thinking about all of the summer students, I really had no idea where I was going to go. I did know that ultimately I was going to do a PhD. I don't know where I learned that, but that was sort of inbred into me. And I knew it was going to be something in life sciences because it just caught my attention. But beyond that, I had no idea I'd work on marine mammals one day. It wasn't my childhood passion. I did watch Flipper as a kid. And I certainly never thought I'd be a professor either. But life has its twists and turns. And the one thing I think I've come to appreciate in thinking about what could I say to you today that might connect is that I've appreciated the role that the unexpected opportunities and the unexpected discoveries have played in shaping my career, as well as how my science education prepared me to handle the unexpected. So how did I end up here anyway? Well, I grew up in Canada's ocean playground. And I think if anybody recognizes where that is, it's because they're also from there. And that's Nova Scotia. And I spent my summers, in fact, my dad was an oceanographer. So I spent a lot of my summers on the beaches exploring life, and he was a physical oceanographer studying ocean currents. I also grew up just loving mathematics and the purity of getting the right answer, which I have to say today in the biology I do, there is no right answer. But nevertheless, I love the purity of math. It ended up taking me to McGill University. And in fact, I planned to study mathematics. And it didn't take too many lectures before I began to realize that there must be more to than just doing math. And so I took the initiative to create my own program as an undergrad. And I proposed to combine, combined major in ecology and mathematics, and looking for a way to apply math skills to solving biological problems. And I know that today that's sort of second nature, but I know at the time at McGill I had to go up to the dean to get permission to combine these two areas. I also grew up with this love of pets and animals, and this is my cat. And as well as growing up reading books. And I'm sure that I can see a few of your professors here that relate to these books. It was the Hardy Boys. I had every one and I read them and I just love these mysteries. Particularly because, just like math, they always end up with solving the problem and the right answer comes out at the end. And there are all some of my favorite titles here: The Secret of the Caves, The Missing Chums, House on the Cliff. And in some ways, I managed to combine some of what I grew up with and enjoyed as a kid into a profession. And I think of myself as doing ecological detective work. And one of the mystery books I've been working on for 24 years has been this mystery of the missing lions. And this has been a big focus of my own research career, has been trying to figure out why in Alaska and Russia stellar sea lions are on the endangered species list, whereas here in British Columbia, going down to California, they are growing exponentially. Our marine mammal populations in British Columbia are so healthy, but you go further north and they're in real trouble. And all we can say for sure about these animals is that they're missing. And we know they didn't just move house, they've not moved elsewhere. But we do have, like in any good mystery, a person of interest. And you can probably guess right off who would that person of interest be. Well, of course, it's the commercial fishermen. I don't think there's a day that goes by that we don't read something in the newspapers about overfishing. And as it turns out, this decline of stellar sea lions, they've declined by 85%. It happened at the very same time as the world's biggest fishery began to grow and grow. And that fishery is for walleye pollock. And I bet if any of you eat fish products, every one of you has eaten walleye pollock. You've eaten the #1 prey species in the sea lion diet where they are declining. So walleye pollock is turned into surimi. So you eat California rolls, that's sea lion food, minus the mayonnaise. And so there is certainly a correlation in time and place. And so we have our person of interest in the same room as where our sea lions went missing. What's really strange in this case, though, is that there are no bodies. The sea lions just aren't there. So it is as though we can do some pathology and figure out what's killed them. They're just missing. There's fewer and fewer each year. So one of the first analyses that I did, and literally looking at about 1992, and I thought this was this open and shut case. I would use my math skills and I would simply do simple correlation type analyses and find out where fishing was the most intense, where they had the most boats. That's where the declines were the sharpest, and therefore you could just restrict fishing. Well, I found the exact opposite. Where the heaviest fishing occurred, sea lions were actually doing better. And where there was absolutely no fishing occurring is where the sharpest declines had occurred. It made no sense. It was an unexpected finding. We also found in trying to look at animals where they're declining that we didn't find any sign of nutritional stress in the sense of seeing malnourished animals. We know how to identify them. Their ribs show through. They're in poor health, aborted fetuses, and various things like this. We weren't finding those sorts of things. And so then we began thinking outside the box about another possibility. And that was that, well, maybe it isn't a problem of catching too much of this fish. Maybe the sea lions are eating too much. So maybe the trouble is that it's not that they're not getting enough pollock to eat, it's that they're getting too much to eat. Now, of course, that's really sacrilege to even suggest such a thing, because the jury has already decided. You didn't even need any scientific evidence to know that fishing was the cause. So how does this work, this overabundance hypothesis? It's been called the junk food hypothesis. And I'll just explain to you simply as you can have too many low energy prey, such as cod and pollock, in the diet, and not enough of the fatty fish, sand lance and herring. So what we know is that before the population declined, before this fishery built up, they were eating mostly high-fat fish. So the dark flesh fish, herring, sand lance, capelin, and they're not getting that same quality of diet. They're eating the lean fish. So for Steller sea lions, junk food is something so the true definition is something that's poor nutritional value, and if you're a sea lion that can't wear a fur coat or burn fossil fuels, eating low-cal food, lean cuisine, is the junk food. I do realize in hindsight I should have called this the low-cal hypothesis or the lean cuisine hypothesis, because junk food has a double meaning when we put it into human context. So maybe to make a bit more sense to that, so how can you understand this? Think of it as if I put you into a field of celery and I go, look, this field, there's 100,000 tons of celery there. Go and eat as much as you want. And you're going to eat that, and I think you all know you're going to come back and say, I'm hungry. And the truth is you'll get full before you have consumed enough calories, and you cannot process enough. Well, the hypothesis we put forward is that this is the same for Steller sea lions, that they can get full eating low-cal food and not get enough calories. And the trouble didn't seem to be for adults, it's really for the young animals, because they require so many more calories per unit body mass to grow, which is the most expensive thing that mammals do. So the question then becomes, so are sea lions living in a sea of celery, where this pollock is effectively the celery of the sea? So I realize I'm trying to summarize 24 years of research into 2.4 minutes. But just to suffice it to say, we did a number of tests to begin with, starting first with the belief that they were starving to death because there wasn't enough to eat. So we did field studies. We've run a very large captive program where I brought sea lions into the Vancouver Aquarium. We run all sorts of simulation models, written ecosystem models, and various sorts of laboratory analysis as well, looking at everything from stress hormones to reproductive hormones, DNA analysis of fecal remains, trying to reconstruct things. So first I just want to give you a quick clip of what sea lions sound like and the sorts of research we've been doing. This is one of our research boats. So we typically survey, this is a non-reproducing site, and we will be collecting fecal samples for, as I mentioned, everything from hormones, stress hormones, trying to look for indications of health problems in the population, diet samples looking for marked animals that we can recognize as well. This is a breeding site, so the young black animals are the pups. We've weighed pups at different sites, and where we find that the pups are the smallest and lightest is where the population is growing. You see a bull in the water. You can see a couple adult females up in the rocks as well. And here we've got, so these big males can weigh up to 2,000 pounds. They're just gigantic. The pups are weighing a hefty 50 pounds or so. And they're learning life skills right from the very start. You've got young males that are fighting. They're just only about a week old and they're already learning how to fight. So in a quick summary, what we found in our field studies in Alaska and British Columbia, because we're comparing increasing to decreasing populations, is that the decreasing populations counter to everything we had expected, because we all believe when I started in this in the 90s that the overfishing had reduced the prey. Well, instead of going really long trips because there wasn't enough food, they went on really short trips. They spent more time nursing their pups. They produced bigger young. And where the population was growing is where they had the hardest time finding food. It didn't make sense, but it does make sense in terms of the junk food hypothesis. This is one of our animals here at the Vancouver Aquarium. We have a lab that CFI provided funding for. And we did this one feeding experiment, and this goes back to 2000. And it's the very first experiment evidence we had that there was something to the ideas we're putting forward about nutritional stress and junk food. So the pre-experiment was to feed the young animals. They're about 2 years old, walleye pollock. They could eat as much as they wanted to eat. And here you can see they were eating about 5% of their body weight per day. We switched them on to pollock, and again we allowed them to eat as much as they wanted to. As soon as they started to play with their food or throw it around, well, dinner was over. And you see they ate a little bit more pollock, and then we switched them back after about 2 to 3 weeks onto herring, which is a higher calorie diet, and you see they ate a bit more again. So what happened to their body weights? We found here that when they're eating the herring, they were gaining about 0.2 kilos per day, all very normal for a young growing animal. But when we put them on to the pollock, we found they lost weight. They weren't able to maintain their weight, they were losing weight. And they didn't seem to be able to compensate by eating more. Our vet at the time said, don't you think your experiment is unethical? And I go, but this is what they eat in Alaska. This is the standard diet. And we did have protocols for cutoff points on it, but we didn't understand why they wouldn't compensate by eating more. We then put them back onto herring, and they started to recover their weight. Doing subsequent experiments, we learned that they were limited by the size of their stomach. And for some reason, people studying marine mammals just assumed they're all gluttons. There are no limitations. But like people, they too are limited by stomach size. So the key here for this junk food hypothesis is stomach capacity and its young animals. Mothers, of course, don't need as much energy to grow when growth is finished. And so they can consume the extra fish and produce these energy-rich milkshakes. The result is that mothers will keep their pups for an extra 2 or 3 years until that pup is big enough and needs less energy, so now it has the stomach capacity to consume the pollock. So the key here, in terms of trying to understand, so what set this whole thing going? Was it fishing that started it? Well, we've been able to reconstruct a lot of the history of the ocean and recognize that in 1975-76 there was a huge change in ocean conditions. We call it a regime shift. The oceanographers documented it, and we picked it up in fishing records and in biological surveys as well. It's not climate change, it's another phenomena, and climate change is now superimposed over these natural long shifts. And stellar sea lions appear to have evolved to maintain their young for longer during periods of time when they have nothing in the store but lean cuisine. So that's for stellar sea lions, and we're still doing various aspects of this, because the reality is that unlike the Hardy Boy books that I used to read, or watching some of the courtroom shows, nobody confesses at the end. And so and there's no nice wrap up to it. You're left with a burden of proof. And in this case, of course, the results don't fit with preconceived ideas, and so it's a much harder sell. And overfishing would have been much easier if that had indeed been the cause. I did my graduate work on northern fur seals, in essence starting with modeling an ecosystem, writing all these programs in Fortran, and later on just focusing on one piece of that ecosystem, which happened to be a top predator. And that resulted in me being sent to represent Canada in the Soviet Union at meetings, sent to Japan, and I was, 22 at the time. And that's what got me into this, was purely by accident, not because I was destined to do it. We've been involved quite heavily with harbor seals. We have in British Columbia, right here off of UBC, the world's highest density of harbor seals in the world. And we're doing research now using a lot of engineering technology and bio-logging, tracking, reconstructing three-dimensional movements down to fractions of seconds. And suspect that harbor seals may be responsible for the poor return of Chinook salmon and coho salmon by eating these very, very tiny little fish. I've been able to study Pacific walrus and go up to the Arctic. And I was interested in trying to find an alternative way to reconstruct diet. So the way that biologists have been studying diets is to shoot the animals and look at their stomach contents. So I thought, well, maybe we could take a hammer and a chisel and collect fecal remains off the ice. And we demonstrated and published a paper showing that we can reconstruct diets from DNA. And it's frozen at, minus 80 degrees on the ice in the Arctic. So it's the perfect way to preserve your samples. But not the most pleasant way as your ice chipping. I remember going to Home Depot looking for chisels, and the guy wants to help me, says, "What do you want it for?" And I go, "You don't really want to know." But you've got to find the tools you need, because you can't buy it from Fisher or some of the other places where you need some of this equipment. We've been involved with killer whale research, trying to understand if the trouble here is related to a lack of Chinook salmon or something else in the system. Been following the recovery of humpback whales. I mean, we now have humpback whales right out here, but they've been gone for 100 years. There's a whaling station set up in Nanaimo that killed every last one within 3 years. And the very first whale watching business was started by the Vancouver Steamship Company in 1904, 1905. And they would take passengers from Vancouver to Bowen Island to go whale watching. All that ended, but they're back now, and we're following their recovery as well. So that's sort of on the professional side, has been doing this. And at the same time, I seem to get phone calls asking me to take part in a lot of things that I never trained for. And I get these unexpected calls. So one of them came from New York City, and they had questions about whalebone corsets. I don't know if anybody here has ever seen a whalebone corset. If you are, you're dating yourself, because they're from the 1800s. And there's a big museum show in New York City being put on, and they wanted to import corsets from Canada to put on display. And so I was asked to go inspect. This is in the woman's house, by the way. And she has this most incredible collection of corsets. And in order to send them into the United States, she has to pass the Endangered Species Act. And because the material inside these corsets has whale material, then you have to make sure they're not part of the endangered species, or her corsets that she's collected for all these years would be confiscated and destroyed. The trouble was that she wouldn't let me cut them open to look at the remains of the whales that were inside. And so I had to come up with a creative way, so I held them up to the light, and I was able to look through and see this with a very bright light. And you can see one is clearly quite dark and one is quite white, creamy. And I had a couple of other samples from known individuals, and we can tell that the white ones we know are from gray whales. It's a white baleen, and the dark ones come from bowhead from the Arctic. And they have different rigidities, and it is able to, essentially it's the ancient plastics of today, except these are biodegradable. And they're used because they gave the right form and different abilities to bend to give the form in the right spot. Anyway, as a result of that, I wrote this affidavit testifying that these were indeed, without having to open it up. Of course, she crossed into the US and they were confiscated. So then I spent 2 days on the phone explaining to the customs agent how I could be so sure what was in it. And eventually I had to give her a whole course in whale biology and baleen and everything else. And these things did get on display in New York City and are now back in Vancouver in her collection. I've also been getting calls lately from the Royal Canadian Mint. And they go, well, why are they calling you at UBC? Well, they sent me a coin. Actually, you can see it's $100. They didn't actually send me the coin. They sent me a picture of it. And I don't know if they're getting ready for inflation to replace the loonie, because this is not yet on the market. But I hadn't realized this before, but they want to be sure that what they're putting out to the public is accurate. Now, you're probably looking at this and say, yeah, that looks really good. I'd like to have that. But I look at it and go, there's at least a dozen errors in this. How can you put this out? And so I'll just give you one example. So has anybody noticed here? I'm sure you've all seen killer whales in the wild. Has anybody seen the back of a killer whale looking like that? Or even look at that shape, it looks like a shark's fin. Well, you can see it's missing the saddle patches. And we go through and I could pick up all these things. And so I was able to go back and forth with the artist until we get the correct drawing done. Here they sent me then, because they liked the first one, they said, oh, how about you look at this one? So then I got the humpback whale. And I looked at this and said, you know, it looks like it's being shot out of a garden hose. I've never seen a humpback whale like this. And look at the baleen grooves. They don't go down to the tail. Like, that's not what it's, but this is what the artist thought they looked like. And so we went back and forth, and next thing you know, we get the water being more natural, although still a bit too turquoise. But we get much closer. So it now does pass the biological inspection. These look pretty good, beluga whales, but again, it's full of errors. Look here at the shape of these flukes. Like, that's not how they look. They look like spades out of a card game. And so going back and forth, then we get to make them the proper shape. And then finally, I got more killer whales. And again, you can see they're missing saddle patches. But what really upset me is that you look at the heads of them, and those are like dolphins. Where's the melon? And so go back and forth, and then we get them put in. Anyway, so far they haven't sent me a single one of these coins in real life. But at least you can be assured now that when you take a piece of money out of your pocket, that it's as accurate as it could be possibly made. And there we go with the final coins. The other thing that often happens is that I get phone calls, and for some reason they get diverted to me, and some of them are really strange. The strangest one I got was from somebody in Sechelt who said, he says, hear me out. I was down on the beach and I found something, and I think this is really important to science. You should know about it. What I found was a duck-billed platypus. I go, that is amazing, because like they only live in Australia. And so I went through with him, like trying to figure out, he was describing it to me, and essentially I have to take him through a whole key to figure out what it was. And then I finally realized what he was actually looking at was the tail end of a beaver. So I had to break it to him. But we get everything from people calling me up and saying they found ambrose, you know, which is this material that is in the intestinal tract of sperm whales. And they just, and you know, these balls of it, they're worth like tens of thousands of dollars, and it's turned into perfume. There's a huge market for it. And so I said, well, send me a picture, because I know like the likelihood is next to zero. And sure enough, what they found was a dried up jellyfish. And you have to tell them what they've been keeping in their fridge, top secret, is just a dead jellyfish. 2 summers ago, it was about this time of the year, and I was sort of thinking of getting ready for classes and what I had to do. And I got a call from a lawyer in Seattle, and she was representing 2 Indian tribes that had been in court for 35 years, fighting the US government to establish their right to fish. And they wanted me to answer what they thought was a simple question, which is, what species of marine mammals occurred greater than 20 miles from shore in 1855? Like, there's no science from 1855. Nobody was out there collecting this sort of data. And so I put her off and said, you know, I'm really busy, but here are some other people you might call. And 2 days later, she called back and said, you really need to help me. These people need help. Anyway, she convinced me, and so I went to one of my graduate students who's a real orchidteer. She just loves whales, and her parents turned out to be historians, which is perfect. So she's already been brought up to know how to dig through the dusty archives, because we had to go find captain ship logs from the 1700s. We had to find some of the accounts of the very first explorers. And this treaty was signed in 1856. The 2 tribes are here in the Olympic Peninsula. You can see here on the coast, so we've got the Olympic Peninsula, the Juan de Fuca Strait and Vancouver Island at the very top, and the Quileute and Quinault Indians. And the treaty said that they have the right of taking fish at all usual and accustomed grounds and stations is further secured to said Indians in common with the citizens of the territory. And yet for 160 some years, we're still debating in the courts, what did they sign? In Canada, our tribes didn't sign treaties, and now we're trying to. But in the US, they signed treaties, and yet it was never really established. And so they felt, the Indians, that they had stories of when they used to fish 20 miles, 30 miles offshore in open canoes. And of course, I thought about this and said, you know, I kind of doubt it, because I was imagining myself having to get in an open wooden canoe without any modern equipment, no GPS, anything. And I thought, you know, like, I couldn't do it, so I doubt others could. Well, how wrong I was. And we did a couple things. First was just trying to document based on modern surveys, knowing that animals have not changed their behaviors. They're still going to occur because they're still driven by the same basic processes. And these were the 12 species that were found out there. We then compared them to what was in the middens. And middens are the garbage piles, essentially the bones and shells. And many places in British Columbia, if you look in the dirt, you're going to find all these shells. Well, that's because First Nations used to live there, and the refuse just piled up. So we looked through the midden evidence, and we found that one of these species accounted for 80% of all the mammal fauna. Over 80%, and this includes rabbits and deer and elk and bear and one of them, and it was a marine mammal. And as you look at this, guess which one it was? It was this little one down here. It was the northern fur seal, a species that I've studied since I did my graduate work in 1980. And so I thought, well, this was really cool now. And so we moved away from whales to focus on seals and fur seals. And on this map here, I just want to show you how far out to sea that they were going to hunt the seals. At least this is based on stories, and it also fits with the biology. But I can't tell you how far that out. You can't see the land, although I was able to demonstrate you could see the tops of a mountain, so you could navigate to get back to land. I had to use basic geometry going back to my high school algebra to figure out how far they could see and how far they could go to return. And it fits with the biology because that's where the animals are. They feed along the continental shelf break. They feed at night and they sleep in the daytime, and they have this jug handle position where they raise one flipper up and they bring their tail up, and it looks like you just pick them out of the water. And what the natives were doing is that they would set sail in canoes equipped with sails. This picture was taken in the 1800s. And they would set sail at about 2 or 3 in the morning. So when they got out there, they would find these sleeping seals and they would spear them. And they would bring back their canoes full of seals. And this was a predictable thing. It's like a Serengeti where you would have about 3 million of these seals swimming by every spring. And they learned over time to go and find them and bring them back. But there was a little bit of a puzzle to this mystery because in the middens, it wasn't just the females, which we know from tracking migrate through there. They also found that there were pups and there were bulls. And that's a bit of a mystery because we know that the bulls from the breeding sites don't come from Alaska. So here's what I'm going to show you is a map showing starting here with the location in Washington where the tribes were killing the fur seals. And these 4 islands are the only known breeding sites in the 1850s where the fur seals originated from. They didn't breed anywhere else in the world. Although if you come in more recent times, we've got 2015, we've discovered there have been some new colonies that have established, one here in San Miguel Island in the 60s, and we know from tags and animals that these animals came from Russia and from Alaska. There's another colony started here, and so they're beginning to spread to new sites. And again, by doing the archaeological digging, as well as some library digging, we discovered that on the Farallon Islands, that there used to be a sealing station that the Russians had set up, except that they killed every last animal, and in 1841 they shut down the station. Well, modern science didn't even know about this. And yet we're now able to establish that there was a separate population. We've now gained insight from this court case about the recovery of animals and how we've got 2 different ecosystems and can make some better predictions for the future of these populations. So last fall or last spring, I ended up in U.S. federal court, the most intense grilling I've ever had on a federal stand. And the court case was really long and it was quite contentious. And the end result was that the court ruled that the western boundary of the Quileute tribe's usual and accustomed fishing ground is 40 miles offshore. And this is really huge because they've been in court for 35 years. And in the end, it was this fur seal evidence that established the rights. Now, they've got no interest in going out to kill seals anymore. They would just like to have a share of the revenue that comes from all these huge, big boats that are fishing. They feel that it's being caught in their territory, and they should have a piece of it. And so fur seals, in the end, carry the day. I just want to share you just a little bit further. I'm going to show you a little clip because at the end of this court case, I was invited with my wife to go, along with a couple of the other expert witnesses, to their village. And they wanted to just say thank you for testifying for them because they also learned more about their history that they didn't know about. So much of it was destroyed. And they expected to lose because they've always lost. But then they won. And so they invited us back and said, okay, now we're going to have a celebration. And it gave me insights into a way of life that I didn't know still occurred. And so here is the celebration that we had with them, with the Quileute tribe. So this has been one of the probably most satisfying things I've ever been involved with. And, I think and say, well, who would have thought that my training as a mathematical biologist would one day affect the lives of people like this? Another thing that's been quite fun for me. And at lunch I sat with Andrew Young, and he asked me, so, what about my balance in life? And I explained, well, I really have it all mixed up because my professional life is mixed in with my personal life. And one of the other things I've really had fun with is building skeletons. And I'm not paid for this, but we moved into a building and I just thought, what a great chance to take some of the things I pick up on beaches and put them together. And so I worked with one of my former students, Mike DeRoos, and we started the Marine Mammal Skeleton Project. And we've got a killer whale, and we've got some white-sided dolphins, and a couple of stellars, and a minke whale. And in doing this, the architects who were designing the Biodiversity Museum got quite interested and said, well, do you think we could borrow your skeletons to go into our new Biodiversity Museum? And I thought about that for a while and thought, I bet I can do better than that. If anyone want to see what we've done here, you might maybe already been in the building, but if you haven't, I just want to give you a little thing. We've got a Pokemon station in there. In fact, I gather there's 2 things there. So from this, it took me onto this project which we call Big Blue, and I realized at this point I did get a little bit in over my head to go and find the world's biggest animal and bring it here to UBC. And just to give you a sense of how big, so here we've got a killer whale and a dolphin. We've got a couple of other whales. We've got the humpback whale and the gray whale. And I've dealt with all these, but I never really visualized just how much bigger a blue whale was. And yeah, I know that now, but I didn't at the time, in all honesty. If I did, I probably wouldn't have done it. And indeed, it's the largest and fastest growing animal to have ever lived on the planet. And it's the largest dinosaur was just half as big. So I'm just going to show you a little clip. The film was made by Discovery Channel, and that's what Ed was referring to. And when they heard I was going to go out to do a preliminary dig after getting the rights to dig up an animal that had been buried for 20 years by the Canadian Museum of Nature, who had one time wanted to put on display in Ottawa until they realized it was too big and too heavy. And so I went with Mike, the student who had been building the skeleton, and we brought his dad, who's a carpenter, and we brought a cameraman. And here's a little clip showing that. Whoops, let's see if I can. This was in November we went. So we had to know what we just got the rights to before we spent a lot of money trying to dig it up. Whoops. I lost sound. Yeah. Fortunately, these little fellows aren't going to get us too far in fertile soil. So we've had a problem with reinforcements. So right now it's a best guess. We'll actually narrow down to, I don't know, 100 to 100 feet or so. So we're going to start. That's 80 right there. Oh my goodness. This animal is 80 feet long. It's buried about 160,000 pounds of flesh is buried here. And so we're just going to pull a few holes here and there and see what we can get over here. I think they want this today and belly down. And we really don't know what we're going to find. It's going to be just bones, nice and clean, which is what I look for. Or whether or not there's the other end of the spectrum, which is a lot of hot flesh. So we've been doing each time to see that they come down. So we've been doing the size of that, so we tap the air. Well, we found the shoulder blades from going the head of this end. We got the outside of the whale. You can see the blubber that's still stuck to the outside. And as we come through here, we're now looking here for the tail end. We've got one of the vertebrae. Most of us have an egg back further out to the road. In fact, So the big shock in this was to discover that it was still covered in blue skin, meat, blubber, and later when it thawed, there was still red blood in it. So the soil was anoxic, and I remember I went back to the hotel that night, and I woke up in the middle of the night with I think the only panic attack I've ever had in my entire life. I thought, what have I got myself into? It was just Mike, me, and his dad. Like, this was so huge. And fortunately, we were able to get the entire graduating class of veterinarians in Prince Edward Island to come. Some brought their parents to help. And we had all this support in the end to dig up and remove what was just this massive animal. We brought parts back. One of the nice things about being at a university is that I don't think there's any problem you can pose without finding somebody that has an opinion, but more important, often a good idea about what can be done or how to solve it. We brought one of the flippers back intact. We took it to the hospital and we did a CAT scan on it. It smelled really bad, but all the staff volunteered their time after hours to come do it. And then I had my own students here debone, and you can see the similarities to the human arm and hand. We had to do a lot of repair work. We brought the animal into the museum and had to raise everything up. At the time, people thought we were building a dinosaur, but of course it was the blue whale skeleton. So how did my science education in the end prepare me to build a blue whale skeleton or to testify in a U.S. federal court, or figure out why steller sea lions have declined, or even how to inspect whalebone corsets? And I think the simple answer is that what it gave me was the tools to access knowledge as well as to solve any problem, no matter how big or how small. And I can tell you with this blue whale, we had nothing but problems. And I think if I hadn't had a science degree, I would have walked away from it so fast. But I knew that we could work things out. And we worked it out, and it's such a great story. And the story is now told in a Discovery Channel documentary that shows all over the world still, but they also show it in the Beatty Museum. And just here to wrap up, so I titled my talk, Expecting the Unexpected from a Science Education, in part because for most students, I think it's not obvious where your science education is going to take you. And I also chose it because of how often it seems that I think the real advancements we make in science and with knowledge come from unexpected findings. So my simple message to you is to learn to embrace the unexpected that lies ahead of you, as well as to be wary when all your results turn out to be just as you expected. So finally here, in closing, I just want to wish you all the best of luck in taking the skills you've learned this summer with you in whatever you do, because I have no doubt they're going to serve you well in resolving any problem, no matter how small or how big they may be. Thank you very much.
Thank you, Andrew. Are there any questions? We do. Chris.
So in the London Museum of Natural History, don't they have a blue whale? Where does a blue whale mock-up?
So they've had a dinosaur which is being removed, and they're going to be putting a blue whale to replace it. And guess who they chose to build the blue whale? My student, Mike DeRoos, who built our blue whale here. You know, I call Mike the Michelangelo of whale skeletons because he breathes life into it. You know, I think I've seen now skeletons in probably almost every major museum in the world, and they all look dead. They're just like flat. And Mike is half artist and half biologist. And so Mike has the contract, so he's going over there shortly to start to build that whale. And so I'm super proud, as I think everybody is here, when you see your students going somewhere. And so, yeah, so Mike's going to be building it. So it's in process. I think it's going to probably take about a year and a half to 2 to finish.
So you say build it, I mean...
They have all the pieces.
The skin and stuff outside.
Yeah, well, they have the pieces, and I think that, I'm not sure if it's one that's currently articulated going to be taking apart to redo. They have an amazing room just crammed full of skeletons. So I don't know if it's taking one of the existing ones or another one that they have in their collection. But I bet it's going to be just spectacular. But it's amazing how many people turn on the internet to find our whales here, or skeletons, because nobody has anything of this quality. I've become this like skeleton snob. And you're looking at, oh, it's not right at all. You know, they have the flippers in the wrong spot. And, you know, we took an x-ray of the flippers, so every bone is in the right spot. And yet I go and I look at these skeletons and they put the bones in the wrong order. You know, it's like they didn't have the picture for a puzzle on how to do it. So I'm extremely proud of what we have here at UBC.
Can you tell us about any missing bones on your way up?
Oh, yeah. So there was one when we were digging up the skeleton. We discovered, no one had told us before, but we discovered chainsaw marks where somebody had cut off one of the flippers. And I thought, oh my gosh, we have an amputee whale. You know, if I'd known this in the beginning, we would have never gone for it. But we're so far into it, and there's all these expectations. And so when I spoke to some of the local people that were with us, they said, well, you know, don't worry. They said, this is pretty common. People would take these cut off flippers and they would nail them to barn doors. And so I thought, so I'm going to drive around PEI with binoculars looking at barn doors, see if I can see, some looks about the right size and shape. And so what we started was essentially a cold case where we put a story in the newspaper, in case anybody remembered seeing something 20 years ago. And sure enough, somebody showed up at this pit. And he walked like extremely nervous all around it, and the conservation officer knew him. So she went up to him, and he was sort of stuttering. You know, he says, "Well, what's going to happen to the person? Are they going to go to jail?" And so we knew he knew something. And once she assured him that they was not going to jail, it was just biologists, and they just really wanted to get it back, he said that he found it on the road. And he kept one of the bones in his house and he threw the rest away in the woods. And so, but he took me back, his name is Junior Petrie, he took me back there and we went through 20 years of leaf growth and all the stuff that had fallen, and he's scraping his fingers going through. And he reminds me of Gollum. Because and he had his precious, and he kept one big bone in his house, and in the end we put it in the truck, and he reached out and he touched the bone and he says, "Goodbye, bone. You're going to go on a long trip now. I hope I see you again one day." And so he'd never been on an airplane in his life, and he works in a lobster canning plant. So in the end, Discovery Channel bought him an airfare to fly him out. And so he was here in our grand opening. And we only stayed the night because, you know, there were lobsters to be put in the can the next day. And then they went back to PEI. But yeah, so we gave it, and in the end, if you go and look at the skeleton, you'll see the chainsaw mark. We could have painted the bone. So the skeleton is its natural color. It's sort of a brownish tinge. And you'll see this very white bone, which is what people think skeletons look like, but of course they've been exposed to sunlight. And we decided, Junior was such a big part of this story, that we left his piece there. So I look at the whale we have in the museum as though she's the librarian of life, the world's biggest animal. And as you come down, and it's free for all UBC students to come in and go and just explore this incredible diversity of life that we have and see it. And you can also see that she's more than just a specimen. She also has a story behind her.
Well, thank you very much, Andrew.